sarcomas. This ranking of tumor subtypes, resulting from the application of our methodology, allows the identification of genes correlated with differentiation and may help identify novel therapeutic targets. Our algorithm represents the first phylogeny-based tool to analyze the differentiation status of human tumors. Cancer research has traditionally focused on the identification of oncogenes and tumor suppressor genes, but in the last decades, it has become increasingly apparent that disruption of normal differentiation is an important component of tumorigenesis. Lack of cellular maturation is now recognized as a hallmark of human cancers, and the degree of differentiation of a tumor is important for diagnosis, prognosis, and treatment. Investigations of hematopoietic malignancies, for instance, have benefited considerably from an understanding of the differentiation hierarchy of hematopoietic cells. The identification of immunophenotypic markers and gene expression profiles correlated with maturation has enabled researchers to map the expansion of malignant cells to particular stages of hematopoietic differentiation. Such characterization has proven invaluable for diagnostic and prognostic purposes, and continues to provide clues for pharmacological interventions. Furthermore, the extent of differentiation indicated by the histologic subtype of liposarcoma is the most important determinant of the clinical outcome for this cancer type. Nevertheless, attempts to categorize solid tumors have proven difficult due to an incomplete understanding of differentiation pathways from stem cells into mesenchymal and epithelial tissues. The classifications undertaken so far have been based on in vitro measurements of genes expressed during the differentiation of stem cells into mature tissue. This data was then compared to expression profiles of different tumor subtypes to identify the maturation stages to which these subtypes correspond. However, such approaches are not yet widely applicable, since the prospective isolation of tissue-specific stem cells has been possible for only a few tissue types, e.g. hematopoietic, mesenchymal, epithelial,